Good morning, children. Today we will discuss about sexual reproduction in flowering plants. That is Chapter 2, Part 2 in our NCERT textbook. Last class we discussed about the parts of a flower. What are the parts of a flower? First one, pedicel, that is the stalk of a flower. Then the thalamus region where the floral whorls are inserted. Then the calyx, units are called sepals. Then the corolla, the units are called petals. Androecium, the units are called stamens. Gynoecium, the units are called pistil or carpel. See that one. Again, we have discussed about the structure of the androecium, that is a male reproductive part of a flower. Composed of the units are called stamens. What is a stamen? The stamen has the structure of the stamen. The stamen has a long, narrow, slender, thin stalk called filamen, and the tip region contains the bilobed two partitions. Bilobed anther is present. So the stamen contains two parts, filament and anther. The bilobed anther is dithecous, and again it is made up of two halves connected by the connective tissue. It has four microsporangia, so it is called tetrasporangiate anther. Then we discussed about the each microsporangium has a sporogenous tissue surrounded by an anther wall made up of four wall layers. These wall layers are produced from the primary cell derived from the archisporial cell, the four layers of anther wall. Okay, now we are discussing about the anther. Anther wall contains four layers are there. The first one, it is called epidermis. Epidermis is the outer single layer of flattened cells which provides the protection. Okay, first layer, the anther wall contains four layers. See that one. First we discussed about the anther. It is a bilobed and dithecous. Then if you are taking the one anther, that it is contains the four layers are present. First one, epidermis, outer provides the protection. Second, it is the endothecium. It is present below the epidermis. It is a single layer, hygroscopic, and fibrous thickenings are present. So now we should see that one. What is the functions of endothecium? Endothecium provides protection to the pollen grains. Second one, these cells are hygroscopic. When they lose water, they contract and cause dehiscence of anther for the release of pollen grains. Now, next we should see the third layer is called the middle layer. These are 2 to 3 layers of cells present between the endothecium and tapetum. They store food materials. Now the last layer is called tapetum. That is the important one. The innermost layer of the anther wall containing the cells with the rich cytoplasm and nuclei is called tapetum. Now we should see that one. What are the functions of tapetum? The tapetum performs the functions during the microsporogenesis. The first function is it transports nutrients to the developing pollen grains. See that one, the first function is transporting nutrients. It is a nutritive tissue. Tapetum is the nutritive tissue present in the pollen grains. It transports nutrients to the developing pollen grains. Second function is it secretes enzyme. What is the enzyme's name? Callase. Callase. Callase enzyme, and different hormones they are producing. Then next function, it secretes oil coating over the pollen grains. Okay, that is called a pollen kit, which protects the pollen from ultraviolet radiation and also which will attract the insects for pollination. Okay, now that one, see that one, the function of the tapetum. Then last function, it secretes special protein to recognize the compatibility. See that one, these all the functions are very important. First function is it giving to the nutrients for the developing pollen grains. Second, it secretes enzyme catalase and hormones. Then it secretes the oil coating over the pollen grains, that is called the pollen kit, which protects the pollen from ultraviolet radiation and also attracts the insects. Then the function it secretes the special protein to recognize compatibility. Compatibility means correct type of the pollen grains. So these are the four walls. Last class we had discussed. See that one, anther wall contains epidermis, endothelium, middle layer of cells, tapetum is present. Inside the tapetum, the microspores are present, that is the male gametophyte is present in that one. So these are the things last class we have discussed. Again we have discussed the microsporogenesis, that is from the archisporial cell, sporogenous cells by mitosis, microspore mother cell, through the meiosis, microspore tetrad, then the microspore or the pollen grains are releasing out. Now, today's class, we should see that one, the remaining parts of that one. Now, we'll discuss about the structure of a pollen grain. See that one, the structure of a pollen grains. Do you know what is the study of pollen grains? That is called the palynology. P-A-L-Y, palynology. P-A-L-Y-N-O-L-O-G-Y. Palynology is the study of pollen grains. Now we will discuss about the structure of a pollen grain, which is the male gametophyte in the flowering plants. If you touch, if you touch the opened anthers of the hibiscus or any other flower, you can see the deposition of yellowish or a white powdery pollen grains on your fingers. Sprinkle these pollen grains on a drop of water taken on a glass slide and observe under microscope. You can see the shape, size, color and design of pollen grains from different flowers. Let us discuss about the development of the male gametophyte. First we will discuss the male. See that one, now we saw that one, the different variety of the pollen grains, how it is developing, how we got that each flower having the different shape of the stamen, different size, different color, and the pollen grain shape and size also different. Now we will discuss about the today's class. Development of a male gametophyte. See that one, last class we stopped the microspore or the pollen grain. From there we are starting now. Microspore is the first cell of the male gametophytic generation. The nucleus of the microspore migrates from the center to periphery. Periphery means side region, and divides to produce large vegetative cell and a small generative cell. Temporary callous wall laid between the two cells and is dissolved by the callase enzyme. See that one? Finally, the pollen grains are the two-celled stage. Now we will discuss about one structure of a pollen grain. Now we will discuss. Okay, first we will see that one, what is the shape of a pollen grain. Normally, the pollen grains are spherical, oval-shaped or triangular in shape. See that one? It is spherical, round-shaped, oval-shaped or triangular in shape and 25 to 50 micrometer in diameter. Shape first is over. Then it is contains two layers are present, namely outer layer is called exine, EXINE, and the inner layer is called intine. See that one? Spherical in shape, outer layer is called exine and the inner layer is called intine. Then we'll take first outer layer. Outer layer exine is hard, made up of sporopollenin. Very important it is. Sporopollenin means it is the most resistant organic material that can withstand high temperature, strong acids, alkalis and enzymes. It has a prominent aperture or a small openings called germ pores where sporopollenin is absent. Pollens can be well preserved as fossils because of the presence of sporopollenin. See that one? Now we saw that one, the structure. What is the shape? Spherical in shape or oval, triangular. It has the diameter 25 to 50 micrometer in diameter and it is made up with two layers are there, outer exine and inner intine. Exine is very hard and made up of with the sporopollenin. Because the sporopollen is a very hard material, if you put in the acid or in that temperature, strong acid, alkali or enzymes, it will affect also. They are not open, that one. It has a prominent apertures in between. Some openings are there, small openings are there. That is called germ pores, GRMPORES. Where that in the germ pores, sporopollenin is absent. Because of that reason, because the sporopollen is made up with the exine outer covering, because of that reason we can preserve as a fossil because of the presence of the sporopollenin. Pollen grains we can long time we can keep it up. Then we will discuss about the second layer. Inside the layer, that is the intine, INTINE, is thin and continuous layer made up of with the cellulose and pectin. That is the thin layers. Exine is very hard, intine is thin layer and it is a continuous layer. Then now the layers it is over. Inside we should see. If it is a 2 or 3 layered having one vegetative cell, means upper vegetative cell that is a large in size, and one generative cell that is a small sized with 2 gametic cells. Gametes are present, 2 nucleus is present. The vegetative cell is large with abundant lot of food materials are present like starch, oils, and also present an irregularly shaped nucleus. See that one? Sporopollenin made up with the exine, intine is made up with cellulose and pectin, contains 2 types of the cells. Inside 2 types of cells are there. And the large cell is called vegetative cell, contains a lot of starch, food materials, starch, oils and an irregular shaped nucleus is present. Then the second layer is second partition is generative cell and it's a small floats in the cytoplasm, contains one nucleus is present. It is almost it is in the corner region, so it is a spindle shaped with dense cytoplasm and one nucleus is present. Usually pollen grains shed at 2-celled stage in 60% of the angiosperms. In the remaining species, the generative cell divides mitotically and 3-celled pollen grains are present.